Yes, welcome. This one is part 3 of nucleic acid, which is very important chapter. So today we deal about the central dogma of molecular biology, which is all about DNA replication, DNA transcription, and RNA translation to protein. So the central dogma of molecular biology includes transcription, translation, and replication, as you see in this diagram. So we are focusing on DNA replication, DNA transcription, and protein synthesis, or known as translation, in deep in this part 3. So DNA can be replicated, and the DNA can be converted to RNA. The RNA can be also reversely converted to DNA. So the science behind DNA replication and the science behind DNA to RNA conversion is known as transcription, and the reverse of transcription is known as reverse transcription. And this RNA can also be replicated by itself, and it can be converted to protein by the process known as translation. Therefore, translation is the process of protein synthesis, whereas transcription is the process by which DNA converts to RNA, and RNA can be converted to DNA. This one is the central dogma of molecular biology. So it's extension of central dogma to include RNA dependent synthesis of RNA in the DNA is this one. It's very important actually. So when it comes to transcription or RNA synthesis, the process of making RNA copy of a single gene in a DNA, and the enzyme used in transcription is known as RNA polymerase, and this RNA polymerase may be RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So the raw material for the new RNA are 4 ribonucleotide triphosphate, and these ribonucleotide triphosphate can be ATP, CPP, GTP, and UTP are there. So as with the DNA replication, transcription proceed from the direction of 5 to 3 prime, then the new base are added to the free 3OH group. So the process of replication or DNA synthesis and the transcription or RNA synthesis are almost similar in different scenarios. For example, in both cases, their synthesis always proceed from 5 to 3 direction. The both require DNA template, and the both contain 3 basic steps of synthesis, that is initiation, elongation, and termination. In both cases, there is the Watson kinetics based pairing rule that for this base pairing, except in case of RNA, there is uracil which substitute what? The thiamine. The only difference is this one. So the process of replication of DNA or DNA synthesis and transcription or RNA synthesis are different in different scenarios. For example, ribonucleotide are used in RNA synthesis, whereas deoxyribonucleotide can be used in DNA synthesis. Only a single strand of DNA serves as a template for RNA synthesis, and it is copied in 3 to 5 direction. And uracil replaces thiamine as a complementary base pair for adenine in RNA. A primer is not involved in RNA synthesis. This is a major difference. In case of RNA synthesis, there is no primer, but in case of DNA, there is a primer. Only a very small portion of the genome is transcribed or copied into RNA, whereas the entire genome must be copied during the DNA replication. Therefore, there is also no proofread function during RNA transcription, and RNA polymerase lacks a parent proofreading 5 to 3 an exonuclease activity here, but this one is mainly found in one DNA replication or DNA synthesis. So the RNA polymerase must be able to recognize the start point for transcription of each gene and the appropriate strand of DNA to use as a template. They also must be sensitive to single strand that reflects the need for the gene product and control the frequency of transcription. So the region of a regulatory sequence called promoter, usually nearby with the transcript gene, control the binding of RNA polymerase to DNA and identify the start point or the point of replication. That is known as OIC in case of prokaryotic, whereas different point in case of eukaryotic. So the frequency of transcription is also controlled by regulatory sequence within the promoters nearby the promoter or promoting proximal element, and by other regulatory sequence such as enhancer that may be located at considerable distance from the start point or the ORIC case of prokaryotes. So both the promoter proximal element and the enhancer interact with protein that stabilize RNA polymerase binding to the promoter. So promoter are a sequence in DNA that determines the start point and the frequency of transcription. So promoter are the point at which that DNA starts or can be located, and the frequency of transcription can be controlled. So in both prokaryotic and the eukaryotic, the adenine and the thymine rich sequence in the promoter determine the start point of transcription by binding to protein that facilitate the binding of RNA polymerase. In E. coli, this consensus sequence is TATATT or thiamine adenine thiamine adenine adenine thiamine, which is known as TATA box or Prino box, or recognized by sigma subunit of bacterial RNA polymerase. So a similar sequence in the eukaryotic gene has also consensus sequence of TATA, also named as Hogeness Goldberg box. This is the name of scientists that identified this promoter for the first time. So in eukaryotics, protein known as general transcription factor or basal factor bind to the TATA box and facilitates the binding of RNA polymerase 2, the polymerase that transcribes messenger RNA, that means. So this binding process involves what is known as transcription factor or TF2 factor for RNA polymerase, and the RNA polymerase 2 binds to the complex of transcription factor and to DNA, and it is aligned to the start for the transcription, that means. So the rate of transcription can be further increased by binding of the other regulatory DNA binding proteins or gene-specific TFF to additional gene regulatory sequence such as the promoter proximal or enhancer region. So this one is different TF factors are there. This one is classes of what promoters that are used for what eukaryotic transcription actually. So I have to see and differentiate each promoters that can be used for transcription factors. So the RNA polymerase binds to the promoter occurs only under some condition when the gene is on, and the gene have actually on off button, the same like your switch. So the RNA polymerase on the promoter occurs only under some condition when the gene is on. So once it is bound to the promoter, the RNA polymerase unwinds off a small section of the DNA and they use it as a template to synthesize an exact RNA copy of the DNA. So the double-stranded DNA has coding and the template strand. The DNA template strand is a strand that is used by RNA polymerase during the process of transcription. So it is complementary and anti-parallel both for the coding or non-template strand of the DNA and to the RNA transcription produced from the template data. So this is how the DNA template can be built and used for what? The replication. Thus, the coding strand is identical in base sequence and the direction to the RNA transcript, except wherever the DNA strand contains thiamine and the RNA contain what? The uracil. So the DNA coding strand and the DNA template strand is, seems like this one. So the winding can be expressed from this 3 to 5 in or 5 to 3 in. So type of RNA polymerase, bacterial cell have a single RNA polymerase that transcribes DNA to generate all of the different types of RNA, messenger RNA, ribosomal RNA, and the transfer RNA. And the RNA polymerase of E. coli contains 4 subunits, that is alpha to beta, beta which form the core enzyme. There is also another protein called sigma factor binds the core enzyme and directs binding of the RNA polymerase to specific promoter region of the DNA templates. So together alpha factor and the core enzyme are known as RNA polymerase holoenzyme, and the E. coli has a number of different alpha factors that recognize the promoter region of different group of the genes. So in contrast to prokaryotic, actually eukaryotic have 3 RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Polymerase 1 can produce RNA ribosomal RNA, the 18 sedimentation coefficient, 5.8 sedimentation, the 28S, and this polymerase 2 produce messenger RNA, and polymerase 3 produces small RNA such as transfer RNA, ribosomal RNA with 5S, and other small RNA. So all of these RNA polymerase have the same mechanism of action, however they recognize different types of promoter actually. So they also differ in their sensitivity to inhibition of alpha amanitin, which is a toxin from the poisonous amanitin mushroom. And the RNA polymerase 2 is inhibited at low concentration, and RNA polymerase 3 can be inhibited at high concentration, but RNA polymerase 1 is quite resistant to this amanitin, which is a poisonous from Amanita mushroom. So RNA polymerase and the DNA polymerase are there. RNA polymerase require primers, but DNA polymerase require primers, whereas RNA polymerase cannot. And RNA polymerase can synthesize 15 N2 per second, but this can be almost 1000. How much it is vary or different from this? So the working principle is from 5 to 3 direction in both cases, and works in both DNA strand in case of DNA, and only a single strand on RNA cases. So this is the difference between the two, and this table shows that how this RNA or polymerase can be sensitive toward this amanitin, which is poison from the mushroom known as alpha amanitin, which is the DNA polymerase 1 is actually, which is insensitive toward this. Here monocistronic and polycistronic messenger RNA are there. In eukaryotic, each gene is transcribed to give a separate messenger RNA in each messenger RNA molecule, therefore encode the formation only for a single protein, that is known as mono, as the name indicates. Mono is single. In prokaryotic, several clusters of related genes known as operon are transcribed together to give a single messenger RNA, which is therefore called polycistronic. So polycistronic messenger RNA yields several protein, whereas monocistronic messenger RNA produces single protein. This one is known as the ratio of the proteins that can be produced by these two messenger RNA. So prokaryotic transcription, especially in bacteria, binding of messenger RNA polymerase with alpha factor for the promoter region of DNA causes what? The two DNA strands to unwind and separate within the region approximately 10 to 20 nucleotides in length. As the polymerase transcribes the DNA, and the untranscribed region of the helix continue to separate, whereas the transcript region of the DNA template rejoin to DNA partner. So this sigma factor is released when the growing messenger RNA chain is approximately 10 nucleoside long, and this elongation reaction continues until the messenger RNA polymerase encounter what? A termination signal or transcription termination signal. So this transcription step is, you know, that initiation, elongation, the termination actually. So in case of initiation, after messenger RNA polymerase binds to the promoter, the DNA strand become unwind, and the polymerase initiate RNA synthesis at the start point of the template strand. That means so elongation is simply increasing the length of what this DNA in the elongating RNA by transcription from 5 to 3 direction. Termination is means simply eventually RNA transcript is released, and the polymerase detached from the DNA. That it means so it is the complete RNA transcription can be expressed at the termination stages. So two types of mechanism for termination of transcription are there: Raw independent termination, the Raw dependent termination. So the RNA transcript must be able to form a stable hairpin loop or turn that shows the progress of RNA polymerase and cause it to pause temporarily. So the tetrameric RNA polymerase can recognize the termination region on the DNA template. That means so this one is a Raw independent termination, whereas Raw dependent termination means involves the binding of protein. That Raw is actually the Greek letter Rho factor, which causes the release of RNA transcript from the template. So as a termination site, this Raw factors displaces the DNA template strand, which facilitating the dissociation of the RNA molecule. This one is a diagram how it can be shown this inactivation of RNA polymerase, especially by using rifampin present. So rifampin bind to the RNA polymerase and change its conformation so that it cannot initiate what RNA synthesis. So rifampin is a drug, but if there is no drug, there is this inactivation cannot be what exists. So inactivation can be done by using what is rifampin drugs. So in the polycystic transcript in prokaryotic is translated as being transcript. So this transcript is not modified in the trimeth, and if it does not contain what intron, the region within the coding sequence of the transcript that are removed before translation can be occurs. So several different proteins are produced during translation of polycystic transcript, one from each cistron. That actually, in prokaryotic, this ribosomal RNA is produced as a single long transcript that is clipped to produce what 16S, 23S, and 5S ribosomal RNA. This S stands for sedimentation coefficient, as we see in the introduction section of this chapter. When you come to eukaryotic transcription, this eukaryotic transcription is more complicated due to the size of the genome and the organization into nucleosome coupled with protein. So in eukaryotes, transcription and replication occur in nucleus, whereas translation occurs in cytoplasm. And in addition to RNA polymerase recognizing the promoter region and initiating RNA synthesis, several supplemental transcription factors bind to distinct sites on the DNA, either within the promoter region or some distance from it. So different enhancer or which are absent in prokaryote are special DNA sequences that increase the rate of initiation of what transcription of RNA polymerase. This RNA polymerase 2 transcript most eukaryotic gene that encodes to protein, and recognition of the promoter and initiation of the transcription by the RNA polymerase 2 require a number of general and specific transcription factors. The different cell types produce different types of protein. Thus, red blood cells produce hemoglobin, whereas white blood cells make antibody, and the enhancer do exactly what their name indicates. As enhancer means what initiating some transcription. It means so they enhance the initiation of transcription as a result of binding to specific transcription factor. So this binding of different factors determine which gene are to be transcribed, and RNA polymerase bind to the transcription factors complex in the promoter region and to the DNA. So the double helix must assume the loss of conformation during that time and transition temporarily from the nucleosome core, and the helix become aligned with the region that starts the point of transcription actually. So the DNA strand separation become what started to occurs. So the synthesis of RNA transcript is initiated, and the RNA transcript is elongated or copying the DNA template. The DNA strand separate as a polymerase approach and rejoin the polymerase phase, and the mechanism by which eukaryotic transcription is terminated is not well understood still now. So scientists can be focusing on how this eukaryotic transcription can be terminated. So what the difference between this eukaryotic and the prokaryotic transcription is that in eukaryotic have extensive processing of primary transcript, particularly the precursor of messenger RNA, which is pre-messenger RNA, and eukaryote also have 3 polymerases rather than just one present in prokaryotic. Eukaryotic messenger RNA usually contain the coding information for only one polypeptide chains, and again eukaryotic RNA is transcribed into the nucleus and migrate to the cytoplasm where translation can be occurs. This one is how eukaryotic can be different from what prokaryotics. Post-transcriptional modification. So different post-transcriptional modification can be takes place in prokaryotics. The transcript messenger RNA is ready to be translated into protein, but in eukaryotics the primary RNA transcript or pre-messenger RNA of the gene needs further processing before it can be translated. Therefore, it needs to transport it out of the nucleus into the cytoplasm. Then after there is different steps in RNA processing. It can be add a cap to the 5' end, add poly, add tail to the 3' end, and removing what the intron and left with exon only. So this 5 capping indicates that RNA is inherently unstable, and especially at the end are modified to protect it. So the primary transcript is capped at the 5' and by 7-methyl guanosine as it is transcript. So the cap can be sealed at the 5' of the primary transcript and decrease the rate of degradation, which can also serve as a recognition site for the binding of the mature messenger RNA to the ribosome at the initiation of protein synthesis. Not only this, it can also help to distinguish messenger RNA from the other types of RNA present in the cell. So the next step is the addition of poly-R9 tail to the 5' as shown in the diagram. The poly-R9 tail can be what the repeated ATP addition and phosphate release at the 5' cap of the end. So this one is the poly-R9 require the poly-R-nylene signal sequence in the RNA that help to direct at the cleavage of transcription, and addition of poly-R9 tail by polymerase can be RNA polymerase that does not reduce or template actually. So this tail is a protein binding site and protects the messenger RNA from the degradation that it means. So the removal of intron is the third step, and rejoining to the exon, that means so eukaryotic many protein encoding gene or called exon are interrupted by intron or intervening sequence which do not code for protein, and the process is known as splicing. The intron are removed from the primary transcript and the exon are joined so that the final mature messenger RNA consists of exon only and the specific functional of polypeptide, and the a complex molecular machine that completes this task is known as spliceope, and the key step in messenger RNA splicing are performed by RNA molecule that rather than the protein that element. So this one is the third step, how intron and remove and exon can be joined. So this one is a diagram how this phenomena can be takes place. Transcription starts at this exon and terminate start at the promoter leg, terminate at the exon that means. So the process of transcription slice out intron and finally translation or protein synthesis can be takes place. So the mature RNA is containing a leader sequence that includes the cap, the coding region comprising exon, and the trailing sequence that include the poly-R9 tail. This mature messenger RNA complex with the poly-R9 binding protein and others protein, it travels through the nuclear pores into the cytoplasm, the place where a protein synthesis in the ribosome can takes place. There is it combined with the ribosome and directs the incorporation of amino acid to polypeptide that means. So when it comes to reverse transcription and RNA replication, according to the central dogma proposed by Francis Crick, formation flow from DNA to RNA to protein, that means transcription, translation, that means. However, retrovirus provides one violation of this rule. When retrovirus invade our cells, their RNA genome is transcribed to produce DNA copy. The enzyme that catalyzes this process is encoded in a viral RNA and is known as reverse transcriptase. This reverse copy integrates into the genome of infector cell, and the enzyme of the host cell are used to produce what many copy of the viral RNA as well as the viral protein, which can be packaged into a new viral particle that is made. So this reverse transcription catalyzes 3 different reactions actually: RNA dependent DNA synthesis, RNA degradation, and RNA dependent DNA synthesis. This one is known as reverse transcription. Some viral RNA are replicated by RNA-dependent RNA polymerase or RNA replicase, and this RNA replicase lacks what separate proofreading of endonuclease activity. And all RNA virus, with the exception of retrovirus, must encode the protein with RNA replicase activity because of the host cell do not possess this enzyme. So this RNA replicase has an error rate similar to that of RNA polymerase, unlike the DNA that RNA polymerase RNA replicase are specific for the RNA of their own virus, and the RNA of the host cell are generated into replicated again. This is the mechanism how retrovirus can be what followed disease reverse transcription phenomena. So this explains how RNA virus are preferentially replicated in the host cell, which contain many other types of RNAs. So this one is the phenomenon of reverse transcriptase. So you know that DNA replication takes place by DNA polymerase, and this transcription takes place when DNA can be converted to RNA, and the reverse is when DNA can be converted, when RNA can be converted to DNA, that is known as a reverse transcription, So retroviral infection of mammalian cell and the integration of the retrovirus into the host chromosomes. This diagram shows that how retrovirus can be incorporated into the host genome so as to change the direction of the central dogma of the normal central dogma of DNA to RNA rather than RNA to the DNA. So that's known as reverse transcription. Mainly takes place in cytoplasm of the cell. So translation, as we mentioned before, it is simply the way of protein synthesis. The phenomenon of protein synthesis is known as translation. The genetic information stored in the chromosome and the transmit to the outer cells through the DNA replication is expressed through the transcription to RNA, and in case of messenger RNA, subsequent translation into the protein. So the overall flow of information in biological cell from DNA to RNA to protein is known as central dogma of molecular biology, and the pathway of protein synthesis is called translation because the language of nucleotide sequence on the messenger RNA or genetic code is translated into the language of an amino acid sequence. The major component required for translation is what all amino acids required: messenger RNA to be translated, transfer RNA, functional ribosomes, energy source like ATP and GTP, enzyme like aminosyl transfer RNA, cyanidase, and the protein factor needed for initiation, elongation, and the termination of the polypeptide chain is very precursor or very important, crucial for translation phenomena. So these protein synthesis and amino acid antibiotics that found that this protein synthesis and antibiotic have some relationship. So what is that? So many antibiotics that are used for combating bacterial infection in human cell can take advantage of differences between the mechanism for protein synthesis in prokaryotic and the eukaryotics. The bacterial ribosome is smaller, which having got 17 coefficient rather than 18, and has different and somewhat simpler complement of the RNA in the protein molecules. For example, if you take antibiotic Streptomycin, which bind to Sartinase ribosomal subunit of the prokaryotic, and which interfere with the initiation of protein synthesis and cause what? Misreading of messenger RNA. Therefore, many antibiotics can perform their function in different scenarios, and some antibiotics can perform their function by inhibiting this protein synthesis or translation. Tetracycline also another antibiotic, chloramphenicol, erythromycin, these all can be antibiotics that inhibit translation. This in general prevent translation step and the movement of peptidyl transfer RNA from adenosine to the phosphate site is what site on the ribosome can be inhibited by these antibiotics. This one is the mechanism of function how antibiotic can be performed their function. Note, there are also agents that inhibit the eukaryotic protein synthesis. For example, diphtheria is a highly contagious disease caused by toxins secreted by the bacterium Cornea bacterium diphtheriae. This diphtheria toxin catalyzes ADP ribosylation of the eukaryotic elongation factor, and thereby inactivating it and then inhibiting the protein synthesis lead to what? Cell death. So these different bacteria can be inhibit what? The phenomena of translation. So protein synthesis and the energy expenditure. So protein synthesis requires a considerable amount of energy, and the formation of amino acid transfer RNA required to high energy phosphate bond, and as each amino acid is added to the growing peptide chain, 2 GTP or guanosine triphosphate are hydrolyzed, and one at that step involving Fi, and the second at the translocation step. So thus 4 high energy bonds are cleaved for each amino acid of the polypeptide, which needs what? A large amount of energy. So in addition, energy is required for initiation of protein synthesis and the photosynthesis from a nucleotide triphosphate precursor of the messenger RNA, transfer RNA and the ribosome RNA can be involved in the translation phenomena. So this one is how translation, the transcription, or the general central dogma of molecular biology can be takes place. Thank you for watching, and until the next video, I will wish a good week for you. And don't forget to share, subscribe, like, and comment on the comment section of the channel for getting the consecutive video or tutorial video, which is very important for your knowledge.